Hello, I'm Laurentius Huber from NIMH in the group of Peter Banditini, and I want to thank the organizers of the symposium, specifically Amir Shmuel, for inviting me to talk about layer fMRI methods to get data in humans. And I have no financial interest to disclose with respect to this presentation. In preparation of this lecture and similarly seen previous lectures, I did check what kind of material is already out there. And I found a lot of great materials from lectures for more than a decade now that all focus on the acquisition and analysis methods of acquiring and analyzing data in humans at the mesoscopic scale. Increasing the resolution with advanced instrumentation provides new insights. And I would like to explain this with this little metaphor from astronomy. So let's look at this beautiful night sky over there, and you see that this spot of the night sky is a bit brighter, right? And when you zoom in, you see it's a beautiful nebula, a cloud. And then when you zoom in, you see there are these subclouds in there. And when you zoom in further and further, ultimately you do hit the resolution limit of Earth-bound telescopes, like for example, La Silla in Chile. And then with decades of methodological improvements, specifically hardware improvements going to space, you can get the same nebula with much more details. You see these contours and little ribbons on these contours, right? And then with another decade of methodological improvements, now you see even more detail, right? You not only see the contours very nicely, but basically also these ripples within the clouds, right? You literally see the cloud evolving and new stars being born. And I like this metaphor because this is just about the time scale of fMRI developments, where the first huge leap forward happened in the early 90s to see brain activity changes non-invasively that kind of are specific to one brain area or brain system and not another one. And then with decades of improvements, resolutions were already so high that you could not only see brain systems, but basically also look at distributions within those systems. For example, how the striate cortex is different from the extrastriate cortex. And then with the newest generation of scanners and methods, you can not only see activities within the gray matter ribbon, but also distributions, how they distribute across different cortical depths of the gray matter ribbon. For example, these little stripes or columnar structures. And these kind of structures can provide a lot of new insights, which I would like to explain in this experimental series now. So here we asked participants at conventional resolutions to move their fingers. And at conventional resolutions, you see these kind of blob of activities in the, for example, primary sensory motor system, which is nice and pretty cool to see. However, then when you ask the person to just think about moving their hand without actually engaging the muscles, you kind of see very similar blobs popping up too. Maybe the skew is a bit different and it's certainly weaker, but overall it can be quite frustrating how similar the brain activation maps look like given the very, very different behavior. So then when you zoom into this tiny patch of the cortex and increase the resolution further and further and further and further, you ultimately start to see these little stripy patterns that I would like to interpret as layers. Namely, we have these voxels at the superficial layers, those ones that are responsible for neurocomputation of integrating a lot of cortical input, feedback, for example, or input from motor planning areas, sensory feedback. Whereas these voxels deeper in the gray matter are the ones that have the bed cells and direct connections to the spinal cords to trigger the muscles to move. This could be understood as the output. Now, at these spatial resolutions, we do see that for the imaginary tapping, we have a very different activity, right? We have only the superficial layers. There's no output. The muscles are actually not being engaged at the end. So now we can see at these resolutions how the brain actually does different things. The same area is involved, but it's involved in a different way, visible at these high spatial resolutions. However, this experimental series here is just one potential application case of how a layer-dependent fMRI can provide new insights for cognitive neuroscience. In fact, in the field out there, including at this conference, there are many other potential applications of layer-dependent fMRI. For example, here looking at directional information flow across processing of visual, like face perception or scene perception, looking at feed-forward versus feedback, or looking at the similarity of the topographical neural activity changes across cortical depth, aka looking at columnar distributions, or also looking at directional connectivity in kind of connectivity networks or resting state. Or you can also use layer-dependent fMRI basically as a vascular filter to learn more about the conscious mechanisms of your given fMRI experiment. for example, with respect to large pile vasculatures, whereas you are more interested in the microvascular responses. And there are many, many more applications of how layer-dependent fMRI can be useful for your neuroscience research. However, when you're using layer-dependent fMRI to ask these kind of questions, we do rely on decades of technical developments. So when you want to do your layer-dependent fMRI, you need to know kind of which hardware to use, then you need to decide which sequence to use and how to optimize it. You need to run your experiment, do your analysis, and then draw your neuroscientific conclusions at the end. And each of those aspects in your layer-dependent fMRI experimental pipeline has been evolved and optimized over decades already. So it has gotten quite fragmented and complicated, to be honest. So you cannot simply change one parameter, like go to 7 Tesla, or just increase the resolution here and assume that everything further downstream will be the same. In fact, you kind of need to have a rough overview of all of these different subdisciplines, because it can get quite complicated. Like, let's look at the most simple case of doing a layer-dependent fMRI experiment. Let's look at an EPI, where we go through a case space by going back and forth and back and forth and back and forth and so on, which is the most simple geometrical pattern you can imagine, right? And I can explain this to my 3-year-old and he understands it. However, when you go to the scanner and you want to optimize this back and forth patterns to go to high spatial resolutions, it's not so easy, because the scanner confronts you with dozens and dozens and dozens and dozens of those parameters. And they're all interdependent, and you need to consider the hardware to adjust them. So it's getting quite complicated. This means that when you want to do layer-dependent fMRI, you need to consider more of those parameters that you would need to consider when you do a conventional standardized fMRI. For example, let's look at a high-resolution EPI, where we do have things to consider. For example, while EPI is the most efficient method out there, it's not efficient enough that you can capture an entire case space of the large matrix sizes that we need in one go before the signal decays away. So we usually only acquire a subset of case space lines, and then we need to fill in missing case space lines by other means. And this could be done, for example, by means of grappa. So here, let's look at a toy model where we have a subsampling by a factor of 2. Then we would have aliasing. And then with grappa, you can unalias those images. However, this comes along with square root of r SNR penalty, because we have less data acquired. But also we have this G-factor noise amplification on top of that. So in the thermal noise dominated regime of these very high resolutions that we are aiming for, it might make more sense to use a different way of filling in the missing case space lines. For example, by combining multiple shots here that are acquired with different prephasers, and then basically not reconstruct data that we have not acquired, but fill them in across different TRs, which means that we have less TRs. However, we don't have the square root of r SNR penalty, which basically accounts for the reduced TR, but on top of that, we also don't have the G-factor penalty. So with respect to TSNR efficiency in the thermal noise dominated regime, it's always more efficient to use segmentation over grappa. In practice, however, it's a bit more complicated, because it also depends on shot-to-shot phase variations. So for example, when we are looking at an 0.8 millimeter data set with a subsampling factor of 6, you can acquire the same data either with no grappa but full segmentation, or with no segmentation and a lot of grappa. And you can see that the extreme cases are not very efficient with respect to TSNR per unit of time. In fact, for a lot of grappa, we are really limited by the G-factor noise amplification. For no grappa at all, we are limited by the physiological noise of the shot-to-shot variations. And usually the optimum is somewhere in between over here. Now, when we go from 0.8 millimeter to an even higher resolution, that we are more thermal noise dominated, then you see that the optimum shifts further towards the left, where it's more beneficial to use very little grappa or no grappa altogether. Aside of the different flavors of doing layer-dependent fMRI in the in-plane direction, there are also multiple flavors of acquiring the data across planes, namely the 2D EPI versus the 3D EPI. While 2D EPI is much more common for conventional resolutions, 3D EPI is much more common at layer-specific resolutions, with about double the numbers compared to 2D in the about 95 papers that have been published since 2020. Anecdotally speaking, 3D EPI was originally implemented simply because people just could not get 2D to work at 7 Tesla with all these off-resonance artifacts and the 3D gradients of those days. Nowadays, 3D EPI is mostly popularized and basically became the standard of layer-dependent fMRI simply because of its higher sensitivity, the higher SNR, and maybe also the more flexible acceleration patterns or sharper slice resolution. But I think mostly it's the higher SNR. Note, however, that 3D EPI does have issues when it comes to like task-correlated motions or phase inconsistencies non-linearly. For example, in this data set where the participants were doing a drinking task and swallowing inside the scanner, where you can see that for 2D, there's basically the center of the brain that moves up and down a bit. And for 3D, on the other hand, it's basically the entire area in the center here is completely like garbage as opposed to the rest of the brain. Without such tasks, however, the image quality that people usually look at is quite decent, and people are usually happy to look at images like that. However, even though this kind of looks okay, when you look at it very carefully, you still see that there are other issues. For example, these very low spatial frequency dark shadows or bright clouds sometimes that I think all layer-dependent fMRI data, 2D or 3D, are currently limited by. And it's particularly visible when you just flip the redirection and look at the opposite of those artifacts. So there is a specific poster about this artifact, namely the fuzzy ripple artifact, also describing how you can account for it, basically doing a dual polarity approach and then accounting those artifacts, getting nice and flat images. So stop by at the poster to learn more about it. I think that most of the improvements within the last decade that made mesoscale imaging in humans much better were mostly coming from software improvements, aka sequence improvements. However, in the last 3 years or so, also new hardware designs were facilitating better layer-dependent fMRI. For example, the development of these dedicated layer fMRI optimized head gradients as part of the next generation 7 Tesla from David Feinberg in Berkeley, which is now also being sold by Siemens more and more under the name of the Terra Impulse Edition. On this hardware, you can achieve 0.39mm fMRI isotropic resolutions, showing all these very fine scale details within the gray matter ribbon, like these different stripes, or along the cortical ribbon, or also perpendicular. For example, maybe indicative of the columns, or also for feed forward or feedback information processing along the ribbon. When whole brain coverage is more of an interest, you can also use this hardware for 0.6mm isotropic layer fMRI connectivity. For example, here using a movie watching paradigm where you can start to resolve these fine scale details in the layer dependent profiles of these subpeaks that can be indicative also of feed forward and feedback information processing. And if you want to use this scanner for your next exciting high resolution project, this is possible because this scanner was just awarded with an NIH U24 dissemination grant. So there is funding for those kind of projects. Aside of the signal acquisition in layer-dependent fMRI, just like conventional fMRI, also in layer-dependent fMRI, we are challenged by not measuring neural activity directly. Instead, we are relying on this neurovascular coupling, schematically depicted here by means of firing neurons. We have an astrocyte and we have an arterial small vessel and a small vein here. So when there is an initial increase in synaptic activity, then there is an increase in energy demands. The vessels are triggered to open up to deliver more oxygen and glucose. The vessels dilate a bit, and a short while later also the oxygenation in the veins changes. And there you already see the big problem, namely that this vein, it's coming from somewhere, right? It might drain neural activity changes as far away as 3mm. And this physiological feature is really a serious problem for many layer-dependent fMRI contrasts, particularly acquisition contrasts that are dominated by these extravascular dephasings, like in gradient echo bold. There has been quite some discussion in the high-resolution community in the last decades to find kind of the best functional contrast out there. And I think there's no consensus about this yet. When you look at the 200 papers that have been published using layer-dependent fMRI in humans, you find that about a quarter of them uses gradient echo bold and basically just hopes for the best. And about a quarter of them uses gradient echo bold, but then also discusses alternative methods that might be more suitable, and about half of the papers out there use a combination of gradient echo bolt and other methods. And these other methods could be something like VASO or 3D Grace or spin echo bolt, ASL, vapor, or something like that. And I think about the spectrum of available methods as you would think about a spectrum of different vehicles. There is no clear winner here, right? Each vehicle has its point and its right to existence, and it just depends on where you want to go and how you optimize your experimental setup to most benefit from the method that you want to use. And very often, it's most beneficial to use a combination of methods, like most of the papers or many of the papers out there do. For example, also, like I would go to a conference. I do not just take the airplane. No, I take the train to get to the airport. Very often when I do feel limited by sensitivity, I do feel inclined to use gradient echo bolt because it has the highest sensitivity, even though the sensitivity does not necessarily refer to the sensitivity of the layer-specific vessels that I care about. It's still the one that has the highest sensitivity. So when I use gradient echo bolt, I sometimes feel like this guy taking the Lamborghini to McDonald's because they worry about food prices. We as the ultra-high field community spend so much efforts in good hardware and high field scanners, so it feels kind of weird to not like burn that sensitivity and put it to good use to make to use a method that's most sensitive to the total microvessels. When I look at the abstract of this conference, the distribution shifts quite a bit. So here, for example, of all the papers in the last decades, as opposed to the layer fMRI abstracts of human fMRI here at this conference, I do see that the vast majority of abstracts uses a combination of methods. In fact, most of them use gradient echo bolt and VASO. And there are two of them that do not use a combination of methods, but then instead try to account for large draining vein effects by other means. For example, with a vascular deconvolution model, or for example, by normalizing vascular reactivity in a kind of division approach. Until now, I mostly talked about the acquisition and hardware challenges of doing layer-dependent fMRI in humans, the early part of this pipeline of doing a layer-dependent fMRI experiment. But the theme that Amir suggested to me for my little symposium lecture was also about all kind of methods, also kind of further downstream when you do your layer fMRI studies in humans, basically covering both methods of acquisition and analysis. So let's talk more about the analysis. And I think one of the most important analysis steps in layer fMRI is the layerification, basically taking any kind of segmentation and converting it into layers to know that which voxels refers to which cortical depth. And at this OHBM, as well as the last 2 years, I think most of the abstracts that actually do layer fMRI or layerification in humans use a method that is implemented in the Laney software suite. So Laney is a package maintained by Farooq Gulman and sponsored by Brain Innovation, and you can give it any kind of patch of tissue type segmentation and it converts it into layers. And you do not need to fulfill any kind of topology requirements of mesh-based layerification, namely having closed surfaces without holes. Another core program in Laney is the column generation by, for example, spanning out local coordinate systems in local patches of the cortex. Here, for example, in the primary motor cortex, where you basically see this cortical organization in this rectangular coordinate system, and you can use this to kind of flatten these patches of cortex and look at topographical representations. Here, for example, mirrored finger representations and how they span across cortical depth. Another very exciting study that just came out is from Alessandra Pizzuti, basically using these local coordinate patches to look at axes of motion representations across columns in the human MT V5, basically flattening the cortex and then looking at different orientations of movement representations as indicated in these little sticks of patches of voxels that span across cortical depth. Another analysis approach in layer fMRI that is being looked at for exploratory purposes more and more are these deveining models, where you try to parameterize the venous leakage, and as the oxygenation changes flow towards the cortical surface, for example by means of an asymmetric point spread function, and then in your analysis you try to account for this venous leakage, for example by a deconvolution model, to tease out these small microvascular lamina specific signals that are sitting on top of these huge unspecific macrovascular both signal changes. When it comes to high resolution, many people worry about motion. I don't, or not too much at least. At the end of the day, it's still fMRI. So we have a lot of independent images, and when there's motion, it's usually not completely constant across all individual time points. So yes, we do lose more TRs than conventional fMRI, and we have a higher dropout rates of not so perfect participants that can lie perfectly still. But for these new scientific studies that we are aiming for, these kind of dropout rates of maybe 20% are not the worst and are not a deal breaker. Most of the retrospective motion correction tools out there, I think, work quite well. And if you tweak them a bit, you can get relatively good inter-run motion correction, as you can see here. So in my experience, just exclusively inviting very good participants and giving them very strong, unfiltered feedback about their performance on top of good head padding can get you a long way. But of course, there are also high-level methodological, also hardware approaches that are worth checking out. I think one of the biggest challenges in layer-dependent fMRI analysis, in my opinion, is the most time-consuming aspect of the analysis, namely the tissue type segmentation. So in my opinion, there is no fully automatic tool out there that provides high quality tissue labels in EPI space across brain areas. Often kind of the cortical ribbon is estimated as way too thin, and some sulci might be completely missing. And it easily takes up to 60 hours of manual labor to correct the first estimation of fully automatic programs of tissue type segmentations. Here, for example, poor Kendrew is really spending 60 hours for an entire whole brain data set just to get an acceptable quality, and has a hand wrist already there. There are very intuitive click-along YouTube tutorials, for example, the one by Farouk Gulban on instruction and tools to really make these manual corrections more efficient, and using also tools to make it kind of semi-manual at the end. And in my opinion, also a very helpful step to facilitate good segmentation in EPI is also to just acquire a structural reference scan in EPI space as well, as for example here by protocols developed by Kenny, where you not only match the spatial geometric distortions with respect to your functional space, because the data are acquired with the same distortions for your structural reference with EPI, but also it basically is just more efficient. You can get a whole brain 0.8 millimeter T1 weighted image like you would get from MP2 Rage in just 2.5 minutes. One further analysis step in layer-dependent fMRI that is getting very popular is the patch-wise removal of Gaussian signal principal components as a denoising step, aka Nordic, by Luca Visioli and Steve Möller, which can tease out signals in even very, very noisy data. Here, for example, looking at a 3 Tesla layer fMRI vaso during a visual motor task. And while you do see that there might be some activity going on, it's kind of hard to see if you don't know where to look for it. And with Nordic denoise, you can really nicely separate this out quite clearly, also with activity in the primary sensory areas. And if you get really, really greedy, you can also turn Nordic up more and more to reject more and more principal components that look like noise-like, until you basically see even activity in the primary motor cortex, where there are very weak signals. However, this comes at the cost of losing interpretability of your effect size as well as the spatial structure to some degree. In cases where you can assume that neighboring columns have similar layer activity, there are software tools out there to allow you to do layer-specific smoothing without signal leakage across layers, which can be really helpful to deal with the high noise level of individual runs. So now we talked about the acquisition and the analysis stages, but I think there are further aspects in layer-dependent fMRI that deserve our attention, because they are important to make the field of layer-dependent fMRI a success, namely the neuroscientific interpretation. For example, one can get the right neuroscientific answer in a very reproducible way, independent of which kind of specific analysis you use. For example, one of the most reproduced features in the field of layer-dependent fMRI is the layer-specific double hump in the primary motor cortex, which you can see with any kind of acquisition analysis, namely with CBF, in CBV, in vapor, in gradient echo bold with phase regression, in 3D grace, or in spin echo EPTI, if you use vertex-based or voxel-based layerification. However, one of the least reproduced feature in layer-dependent fMRI are simple things like attention effects in B1s, where, for example, some people see mostly an effect in the superficial and middle layers, some people see strongest modulations in superficial and deepest layers, some see the strongest response in the superficial layers only, some see an effect, but it's pretty much identical across all cortical depths, seeing no layer segregation. Some people see most strong effects in the deepest layers, and some people see the only significant effect in the middle layers. So I think aside of all these fights in the field and discussions on how to optimize your acquisition and analysis methodology, I think we also should put some attention on how to interpret the data and how we generalize our conclusions from these very specific experiments that we do with the very specific control conditions of that experiment to these general concepts like attention in V1. Now, to give you an overview of the field of layer-dependent fMRI and see where it's going, let's look at some statistics. So here, for example, I'm showing you the number of human layer-dependent fMRI papers, and you can see that it took quite a while to really take off for the field until 2015, where it's really starting to explode. And I'm also quite happy about the development that it's not just a methods field anymore, but since 2018, it's really the applications that drive the field. So there's more neuroscience applications of layer-dependent fMRI than there are methods developments. I'm also happy to see that there are more and more brain areas being investigated. So the first decades were mostly focusing on the vision aspect of neuroscience, but now there are also motor, sensory, auditory, and associate areas, more cognitive areas being investigated too. Another aspect in the field that I think is going in the right direction is, for example, when it comes to data sharing. So until 2019, almost nobody made their data publicly available. And since 2019, at least about a quarter of the papers out there made their data directly publicly available. But we still have a way to go as a field there. Another aspect where we do have a way to go is maybe diversity. For example, when it comes to vendors, layer fMRI in humans is really dominated by Siemens, and this might not change a lot in the next few years, I assume. But also with respect to geographic diversity, there are weird aspects there. For example, most of the scanners are coming from America and Asia. However, when you look at the layer fMRI papers and method developments, it's mostly dominated from Europe. So there is this kind of mismatch there. But even more alarming is the diversity across genders. So across all the layer fMRI papers out there, only 1 out of 4 papers is coming from a female authored citation. And even more alarming, when you look at all the references of all the layer fMRI papers, only 1 out of 7 references goes to a female authored citation, which is really something that we as a field need to work on. Independently also, we as a field, I think, should not forget our climate footprint. So we learned, for example, quite recently that 7 Tesla scanners actually consume quite a lot of energy. So a single 7 Tesla scanner consumes as much energy as about 60 to 100 households, 4-person households or freestanding houses. That's a lot of energy. If you want to stay informed in the field of layer-dependent fMRI, please join us, for example, via the Layer Dinner group. For example, by just downloading this example data set and show the field on how you do your analysis from the raw data to your layer profiles at the end. This Twitter handle is trying to keep you posted about ongoing developments in the field, papers or events. And there are 2 exciting events coming up. For example, in October, there's a 4-day educational course in Boston about kind of teaching the basic principles of layer-dependent fMRI. Because I think many people want to do it, but just don't know how just yet. There's another 2-day layer fMRI workshop. It's more kind of hands-on analysis workshop as part of the ultra high field workshop in Minneapolis later in the same month. And with this, I thank all my colleagues and I thank you for your attention.